Okay friends, here we will see the significance of enzyme kinetics and why we require enzyme kinetics at all for understanding how the enzyme behaves. So let's look at that. According to the Michaelis and Menten, they provided a formula for the velocity for the enzyme activity, that is V0 equals Vmax into the substrate concentration divided by Km plus substrate concentration. Now if you provide a direct plot of it, we will get a straight line where the substrate in x-axis and the velocity in the y-axis. We will get the graph, and the graph showing that the concentration with the activity of the enzyme increasing with the substrate concentration, and then it gets linearized. And if we look at the double reciprocal plot, we also saw this plot is better for getting the idea of both Vmax and Km, and that's what the beauty of the Michaelis-Menten equation. We used Michaelis-Menten equation to find out the Vmax and Km for any chemical enzymatic reactions. Here Km act as the affinity of the enzyme with the substrate, and Vmax on the other hand dictates the maximum velocity of the reaction. Now if you look at the direct plot, we will see two different stages. First, when the exponential increase of the activity is the first order reaction, and then secondly, when the activity of the enzyme is saturating, it's known as the zero order reaction. Now for the double reciprocal plot, we can also get to know about the different types of inhibition, and for different types of inhibition like competitive inhibition, non-competitive inhibition and uncompetitive inhibition, we will have different types of plot that will help us to understand and identify the type of inhibition that is going on. So all of these graphs have their own importance. Now the Vmax will be also beneficial to understand another concept known as the idea of K3, that is the rate constant for the conversion of enzyme substrate complex into the product. And this K3 will be helpful to get us the idea about the kcat, which is also known as the turnover number. Now the kcat can be equals to K3, K sub 3 in some cases. In some other cases, kcat will be equivalent with the value for K3 divided by Km. Now this turnover number is simply depicted by the activity unit, that is how much of the substrate is converted by the enzyme per minute. And we can also get the value of specific activity, that is the unit of the enzyme divided by the concentration of the enzyme. Now this maximum velocity and affinity with the substrate is two major importance and features of the enzyme that are involved. Now the bi-substrate reaction also follows the Michaelis-Menten equation, but one of the substrates should be saturated when they eliminate the other. That's what we will know as the process will go on. Let's look at the significance of this enzyme kinetics. The primary significance of this Michaelis-Menten formula, if we write it down, V0 equals Vmax into substrate concentration divided by Km plus Vmax, Km plus S, sorry. Now this Vmax or Km of the enzyme was obtained by this Michaelis-Menten equation, and this Km and Vmax are graphical, it's not something only theoretical. And this Km and Vmax can introduce different behavioral pattern of enzymes. So if we look at here the two situations, the simple plot of the substrate concentration growing from low to high, and we look at the activity and the velocity reached at the beginning, it's behaving like a first order, then while at the end when it's getting round off, it's the zero order reaction. And if we carry out the same experiment in a fixed substrate concentration, no change in substrate concentration, we will find the graph for different enzymes behaving differently. So fixing a substrate concentration will also help us to identify the effectivity of an enzyme of converting that substrate. So catalytic rate increases with increasing substrate concentration. Okay, that is the first response. Now at very high substrate concentration, and substrate concentration will also affect the rate, that is the zero order part. Therefore, if fixed at a very high concentration of the substrate, in that case the reaction rate and the response contained in a liquid is proportional to the amount of enzymes. If the substrate is varying, then we can tell this is mostly depending upon the substrate, that enzyme, and how the enzyme is going to behave. But if the substrate concentration is fixed, then increasing and decreasing the enzyme content can tell us about the ability and effectivity of that enzyme. Now, for example, here we'll see for enzyme 1, enzyme 2 and enzyme 3, they have a different way of interacting and different way of reaching the saturation point, and that is proportional to the enzyme concentration if the substrate level is fixed at very high concentration. Now if you provide a very low substrate concentration fixed, in that case it will be again a problem, because higher substrate concentration is required for the enzymes to work properly. Now this Km and this S, there is a relation between the two. What is the relation? V0 equals Vmax into K, you can write it. So K3 into K. So what does this equation mean? We will see that in the next slide. Let's look at that in the next slide. Let's describe a little bit more about the Km, that is also known as the affinity of that enzyme with its substrate. Now if you set the Km of constant, it's not, but it's just the substrate concentration when the half the maximum velocity is achieved. So we take this formula of Michaelis and Menten, and if we think, if this V0 equals Vmax by 2, that is the half of Vmax, and put the value of the Vmax by 2, which is the value of V0, into the Michaelis-Menten plot, what we will get is very simply Km plus S equals 2S, which will give us Km equals S, or substrate concentration. So Km will be equal to the substrate concentration when the V0 equals half the Vmax. So when half the maximum velocity is received or achieved, whatever substrate concentration is there will be equivalent with the Km. So high the Km to reach the highest rate, and adding the substrate concentration is also higher, which indicates the affinity of the substrate and enzyme are not very good. If you look at here, let's explain it with a graph. If you look at this graph, when we are using different substrates, this is say substrate 1 for substrate number 2, and go for substrate number 3 with this blue. Now what we are looking at the Vmax, that's a maximum, and that's kind of conserved for all of them same, but the Km are different for different substrates. So what you can see, the Km value will be changed. In some enzymes, the substrate concentration requires to reach half the velocity is less, but for others the substrate concentration requires to reach half the velocity is more. So it's better if an enzyme reaches the half of the maximum velocity in very low substrate concentration. In that case, that enzyme is interacting with the substrate in much greater affinity. That's what we're talking about, the Km, the value for Km. So here the affinity are different for S1, S2 and S3, and here we can say the affinity for S1 is the highest, S3 is the least. Okay. Now if you look at an example to describe Km, that is hexokinase, an enzyme that creates glucose 6-phosphate from glucose. It utilizes ATP and transfer a phosphate group to the glucose at the 6th carbon position. So in this case, this hexokinase can act for multiple molecules or multiple carbohydrates. Actually, it can catalyze several 6-carbon sugars like glucose, allose, mannose, etc. But 3 of the Km vary considerably. Hexokinase is the same enzyme, but depending upon the substrate, you see they start varying the Km. Okay. So if you look at here, for the glucose and mannose, they have a greater affinity. So the value for Km is low, so they have a better affinity for that part. So here you see for this one, and if you look at here, the ratio of this 3 monosaccharide molecular structure, the carbon 3 was found above the 3-dimensional formation, that is the hydroxyl group, has a great influence of the hexokinase glucose performance. Okay. So this is going to tell you about a single enzyme, if it behaves and interacts with different substrates, may have different Km. So the value for Km is not universal. It depends on the type of substrates and enzymes are involved to work with. Now let's talk about another important concept, that is the turnover number, or known as kcat. And again, to understand kcat, we need to go back to the Michaelis-Menten equation and the simple idea of how enzyme and substrate are linked to produce product. We know enzyme and substrate will involve in interaction. They will form the transient state or enzyme substrate complex. Then the product will be formed and it will be taken out, and enzyme will be also being freed. Now if I look at here, when the substrate quality maximum, Kn can be ignored. Now in that case, like let's say the V0 equals Vmax in those cases of very high substrate concentration. And if you look at this equation, you will see when enzyme and substrate is converted to ES or enzyme substrate complex, it's a reversible process with K1 and K2 as a rate constant. And when the ES is converting into the E plus P or product, there is the third rate constant, that is K3, and that is the time that determines the type of activity of the enzyme, because that's the most important part of producing, and this is irreversible step. So what we know is that when the substrate is excess, then this K3 is the most important rate constant that acts like a turnover number, also known as TON. Now turnover number is the amount of substrates converted to the product per unit time that will be depicted here. Okay. In that case, K3 becomes the major controller, the performance of the enzyme, but it depends on when the substrate concentration is very, very high and it's excess. But when the substrate concentration is low, relatively low, in those cases, if you look at the process of Michaelis-Menten equation again, so in that case a Vmax that can be achieved here is the K3, and the association of K3 with the enzyme concentration as well, it provides us the value for the catalytic activity, and that becomes K3 by Km. Km requires the substrate specificity and K3 is the rate constant, and in this case the turnover number becomes this K3 by Km, that's the value. Okay, because in this case the velocity will be K3 by Km Es. So the response rate for the enzyme substrate control of 2 is a second order reaction, and the constant is K3 by Km. Okay, so this is a pointer on a substrate of an enzyme catalytic ability, that K3 as well as the representative of the affinity Km, both type of them represents the process of how an enzyme behave and interact. But while the concentration of substrate were excess, K3 and Km, both of them does not require to work well there. Only K3 can be behaving like a kcat there. So for summary or summarization of this part, I can tell, if you're looking at the first order reactions when the substrate concentration is excess, kcat will be equals to K3. But when there is a second order reaction and substrate concentration is very low, kcat will be equivalent with the K3 by Km, and because in that case substrate specificity by the enzyme is also important, so as the concentration of enzyme as well as substrate both. Let's talk about the turnover number of different enzymes to give you an idea about the process. Let's say this is the list of enzymes, substrates and their kcat values. See the enzyme catalyst, substrate is hydrogen peroxide, we have the kcat values, and the kcat values are given in it's actually given by second inverse, so the amount of enzymes converted into the product. So here this constant K3, it controls, and a control is gathered in the production rate, and the enzymes that finally converting its substrate into the product, and the time taken to do that, because that's obviously depending upon the velocity of the enzyme activity as well. That's also an important part. The unit is second inverse, as we see. Now in this case, if we take one example, that is Rec-A protein, which is an ATPase protein which utilizes ATP as a substrate, it has a turnover number of 0.4, indicating that it takes 2.5 seconds to use a molecule of the enzyme ATP and convert them and utilize them for the association step. So it take ATP, it will take 2.5 seconds to use that molecule of ATP and convert it to ADP. So the number of product transformed from substrate by one enzyme molecule in 1 second will be known as the turnover number. It is the determinants by the affinity of the enzyme towards the substrate. It also is depicted by the velocity of the substrate, and which will also be controlled by either substrate concentration, enzyme concentration, and how the substrate is involved with interaction with the enzyme. Now if we talk about we talked about two types of kcat activities there. Now if you look at the chymotrypsin, it has distinct kcat by Km to different substrates. That's what we need to know, because in varying substrates the value for kcat by Km also changes. Now if we look at here, this is let's say the chymotrypsin, which has a specific R group that will involve it. Now if we change that R group, there are multiple types of amino acids can be present in that R group, like glycine, which only has a hydrogen in the R, norvaline or norleucine or phenylalanine. Now if we change this R group, the behavior of this enzyme also changes. Okay, the larger the group is in this case, the better the catalytic activity according to this picture. Okay, enzymes will have different performance on different substrates, but chymotrypsin prefer larger amino acid groups and preferably aromatic groups. The enzymes of this success catalytic first needed the substrate collision, and both this collision rate they decide, it's also depending upon the diffusion rate, but how those substrate and enzyme collide, this collision depends on how exactly they will be involved in the interaction, and that also determines by the specificity with each other. So you see the value is being changed with time, it's being increased with the bigger and aromatic ring. Now what are the activity units for the enzyme? If you plot the data of reaction time over the product, a formation of product with reaction time, and we have different amount of slope, and if you form a tan with the slope values, we'll get the xy, and the tan theta, that's the formation of y by x according to the picture. So the substrate concentration over the time and velocity is a p divided by minute per minute. The product p means the product concentration in this case, and the unit will be micromole per minute. And what is a specific activity? Specific activity equals the value for activity units divided by the protein concentration and the amount of the concentration in milligram. So determination of the enzyme activity is in the determination of the resulting product per unit of time. As shown in this figure, if the product expressed as micromole catalyzed by enzyme per minute with 1 micromole known as active unit. Okay. So if you like this video, please hit the like button, share this video and subscribe to my channel to get more and more videos like that. Remember, sharing is caring, and it also helps us to promote so that more and more students can watch this and can get benefits just like you did. Okay, and feel free to comment about the video, and also if you think there are some part that we want to talk and we need to talk about and make videos about, you can also share your ideas. Thank you.